Hello everyone. Today I am going to be talking about chlamydia. So Chlamydia trachomatis is a gram-negative bacterium, which means it has a cell wall that appears pink when stained with the gram stain. It is an obligate intracellular pathogen, meaning it can only replicate and survive within the cells of its hosts. Chlamydia lacks peptidoglycan, a crucial component of the cell wall found in most bacteria, making it unique in its structure. Chlamydia trachomatis has several serotypes or strains, each with varying levels of virulence and clinical presentations. One of the main attributes of this bacterium is its ability to infect and target epithelial cells, particularly in the genital tract, leading to the commonly sexual transmitted infection, chlamydia. It has evolved specific mechanisms to enter host cells, manipulate host cellular processes, and evade immune responses, which contributes to its pathogenesis and ability to cause infection. All right, so transmission and epidemiology. Chlamydia is primarily transmitted through sexual contact, including vaginal, anal, and or oral sex. It can also be transmitted from infected mother to newborn during childbirth, leading to neonatal conjunctivitis and pneumonia. Chlamydia is a commonly sexually transmitted infection, which affects both men and women. Populations affected are adolescents and young adults aged 15 to 24 are at higher risk because of increased sexual activity. All right, some statistics about chlamydia. Higher prevalence is observed in low and middle income countries where access to screening and treatment may be limited. About 4 in the US in 2018, there were reported about 4 million chlamydia infections. Chlamydia is the most common among the group ages 15 to 24. Chlamydia is the most reported bacterially sexually transmitted infection in the United States. And just a note, we really can't account for all the chlamydia infections because most people with infections have no symptoms, so they don't get tested or seek treatment. More stats. So 67% of new infections occur among youth aged 15 to 24. 1 in 20 sexually active young women aged 14 to 24 has chlamydia. And then chlamydia rates for African Americans were 6 times that of white Americans. All right, so some chlamydia prevention and treatment. So obviously using a condom, getting tested, and then as well getting on treatment once you have that positive test result. The drug regimen is erythromycin 100 milligrams orally, single dose, or doxycycline 100 milligrams orally, 2 times a day for 7 days. And then first line of treatment is usually Rocephin, 500 milligrams intramuscularly. It's a single dose injection. Alrighty, so some symptoms of chlamydia. So most common symptoms include abnormal genital discharge, pain or burning during urination, rectal pain or bleeding if engaged in anal sex, and testicular pain or swelling in males. Chlamydia can also be asymptomatic, meaning individuals may not experience any noticeable symptoms. So if left untreated, it can lead to complications such as pelvic inflammatory disease in females and epididymitis in males. Other possible symptoms include abdominal pain, bleeding between menstrual periods, and conjunctivitis if exposed during childbirth. That's why routine screening and early detection is critical to prevent long-term consequences and reduce transmission rates. All right, so the culture and diagnosis of chlamydia. So the nucleic acid amplification test is the most sensitive and specific test for diagnosing chlamydia. And infections. They detect genetic material, DNA or RNA of the bacterium in the patient sample. Samples can be collected from various sites, including urethra, cervix, vagina, rectum, throat, depending on the suspected site of infection. NATs can be performed during using urine swabs or other specimens. Another test is the nucleic acid probe test. These tests detect genetic material of chlamydia using specific probes. They are less sensitive than the gnats, but they're still used in some settings where the gnats are not readily available. Another testing possibility is cell culture, though it's less common used because of the more sensitive gnat tests that are just a lot better. So the culture can involve growing the Chlamydia trachomatis in a laboratory setting, and it can provide valuable information about the viability of the bacterium and may be useful in research purposes. All right, so mechanism of the infection. All right, so Chlamydia trachomatis has a unique intercellular life cycle that enables its pathogenesis. The infection starts when the elementary body, the infectious form of the bacterium, attaches and enters the host epithelial cell lining of the genital tract. So once inside the cell, the elementary body differentiates into the reticulate body and the non-infectious replicating form. The RB multiplies within a specialized compartment called inclusion, avoiding host immune detection. All right, some virulence factors. So adhesions, Chlamydia trachomatis expresses adhesions on its surface, such as major outer membrane protein and the adhesion-related molecule PMPS, allowing it to adhere and enter host cells. This is a crucial step in establishing the infection. And then secondly, type 2 secretion systems. Chlamydia secretes proteins via the type 3 secretion system, and which functions as effectors. These manipulate host cells processes promoting bacterial survival and replication. Some effectors inhibit host cell apoptosis, which while others modify the host endocytic pathway to prevent lysosomal fusion with the inclusion. And then there's the A protein. This protein is essential for the formation of the inclusion, the intracellular compartment where chlamydia replicates. It prevents the inclusion from fusing with other cellular compartments, shielding the bacteria from host immune responses. And then lastly, liposaccharides. Chlamydia produces liposaccharides as a component of its outer membrane, which can stimulate pro-inflammatory responses. However, chlamydia's liposaccharides lack the typical endotoxin activity, which allows the bacterium to evade strong inflammatory reactions and avoid immune clearance. Right, so pathogenicity. So the unique intercellular life cycle of chlamydia and its ability to avoid the host immune response and contributes to its pathogenicity. It can persist and replicate within the host cell, leading to chronic infections. These chronic infections may cause chronic inflammation and tissue damage, especially in the genital tract, leading to complications like PID in women and tubular factor infertility as well. Chlamydia trachomatis employs several strategies to avoid the host immune system by residing within the inclusion. Chlamydia avoids exposure to immune effectors in the cytoplasm. It also downregulates the expression of major histocompatibility complex molecules of the infected cells, reducing antigen presentation of T cells. All right, and then this is host immune invasion. So, like I said, it resides within inclusion, chlamydia from immune effectors in the cytoplasm. And yeah, this is what I was speaking about. And then the downregulation of the major reducing antigen presentation into the T cells. That is the host immune invasion. All right, and those are my citations. Thank you.